I want you to try a visualization exercise with me. It's a bit of a cliche, but stick with me because we're going to break the cliche pretty quickly here.
Yeah,
Picture an 80-year-old walking into a room.
I picture it.
Really visualize it. You are likely picturing a specific gait right. Maybe a bit of a shuffle, a hesitation before they speak. If they tell you a story, you might expect it to loop around a few times, maybe lose the timeline, or struggle to retrieve a name they knew yesterday.
Right, yeah. We have this culturally embedded script for what 80 looks like. It is a script of subtraction. Less speed, less memory, less processing power.
Exactly. Less. And that makes sense to us intuitively, right?
It's the default biological assumption. Entropy. Things wear out over time. The lights dim.
Right. But now I want you to take that image and completely delete it. Just wipe it out. Replace it with an 80-year-old who walks in and can recall a list of random words better than a 25-year-old. Someone whose attention span isn't just, good for their age,
Yeah.
but is biologically indistinguishable from a healthy adult in their prime.
And we should clarify, we aren't talking about outliers who are just spry. We are talking about a biological anomaly.
Yes, we are talking about SuperAgers. And for a long time, I think the assumption was that these people were just genetic lottery winners. They picked the right parents, they had the clean genes, and they just coasted. But we're looking at a stack of research today, including a massive study published just days ago in February 2026 in the journal Nature that suggests something completely different.
It really does. It suggests that our fundamental understanding of cognitive decline as an inevitable slide is just wrong.
That is a huge claim, fundamentally wrong.
I don't use that phrase lightly. But when you look at the data we have from the Mesolom Center at Northwestern, combined with this new single-nucleus RNA sequencing data, you have to conclude that cognitive decline isn't just about damage. It's about the failure of repair. And SuperAgers, they are master repairmen.
So today, we're taking a deep dive into the SuperAger paradox. We're going to look at why some brains full of Alzheimer's pathology, literally full of the disease, don't get dementia. We're going to unpack the neuron factory that is apparently running double shifts in these people. And we're going to talk about the wobble.
Oh, the wobble is my favorite part. It's so actionable.
We will definitely get there. It's totally fascinating. But first, let's establish the baseline here. You mentioned the Mesolom Center at Northwestern. This isn't a new project, is it?
No, and that is why this data is so incredibly valuable. You can't just find a SuperAger on the street and study them for a week and learn anything meaningful. You need longitudinal data.
Right, you have to track them over time.
Exactly. The Mesolom Center for Cognitive Neurology and Alzheimer's Disease has been tracking this specific cohort since roughly 2000. So we have over 25 years of tracking.
And to even get into this club, the barrier to entry is incredibly high, right? You have to be over 80. And you have to pass this brutal memory test.
Yeah, the delayed recall test.
Explain how that works for people who might not be familiar.
Sure. So they read you a list of 15 unrelated words, things like drum, curtain, bell, coffee, and so on. You listen to the list. Then they distract you. You have to go do other cognitive tasks for a while.
So you can't just repeat them over and over in your head.
Exactly. 20 minutes later, they ask you to recite the list from memory.
I feel like if you asked me to do that right now and I am nowhere near 80, I would struggle to get half of them.
That's actually the norm. A typical 80-year-old, even a very healthy one, might recall 5, maybe 6.
Okay.
To be classified as a SuperAger, you have to recall at least 9. But the wild thing is, many of them hit 12, 13, or all 15 words.
All 15?
Yeah. They are performing at the level of a 50-year-old and often outperforming people in their 20s.
That is insane. And this isn't common, right?
Very uncommon.
Yep.
Less than 10% of the general population qualifies as a SuperAger.
Okay, so we have this elite group. The assumption for years, as you mentioned, was their brains must be pristine. If I look at the brain of a SuperAger, it should look like a new car, right? No scratches, no dents, no rust.
That was the resistance hypothesis.
Yeah.
The idea was that these people somehow physically resisted the aging process entirely. Their bodies just fought off the amyloid plaques and tau tangles that cause Alzheimer's.
Amyloid plaques and tau tangles. We hear these terms constantly when talking about aging and dementia. Just give us the 30-second refresher for the science fans listening.
Right, so think of them as the hallmark gunk of Alzheimer's disease. Amyloid plaques build up between the nerve cells, kind of like trash piling up in the streets, blocking traffic.
Okay, trash in the streets.
And tau tangles build up inside the cells themselves. Tau is normally a protein that helps stabilize the internal structure of the neuron.
Like the scaffolding.
Yes, but in Alzheimer's, that scaffolding collapses and gets tangled up. So it's like the internal wiring of the house getting completely knotted.
Got it.
For 30 years, the amyloid hypothesis has basically been the Bible of neurology. The theory was simple. The more plaque you have, the more dementia you have. It was seen as a direct linear relationship. Or so we thought.
And this is where the 25 years of Northwestern data just threw a massive wrench in the gears.
A gigantic wrench. Because they didn't just test these people while they were alive. A crucial part of this longitudinal study involved post-mortem analysis. These SuperAgers graciously donated their brains to science.
Which is such an amazing gift.
It really is. And when the neuropathologist looked at the tissue under the microscope, they found something that completely shocked the field.
Some of them were clean. Some actually fit that resistance model you mentioned.
Yes, that is true. Some were pristine. They had completely resisted the pathology. No plaques, no tangles. But a significant number of them, people who remember had the memory of a 25-year-old until the very day they died, had brains that were absolutely dirty.
Define dirty in this context.
I mean, they had high densities of amyloid plaques and tau tangles. If a pathologist looked only at the slice of their brain tissue without knowing who the person was or how they acted in life, they would diagnose them with Alzheimer's disease on the spot. We are talking break stages 2 or 3, which is a moderate level of pathology.
Wait, hold on. This completely contradicts what you just said about the amyloid hypothesis. If they have the trash in the streets and the knotted wiring inside the house, why aren't they forgetting their keys? Why aren't they getting lost or forgetting their grandchildren's names?
That is the multi-billion-dollar question in neuroscience right now. This is the mystery of resilience. Resistance is avoiding the damage entirely. Resilience is taking the damage, having the pathology, but functioning perfectly anyway.
So the hardware is broken, but the software is somehow running fine.
Or a better way to look at it might be that the hardware is damaged, but there is a repair crew working so incredibly fast that the person never notices the glitch.
And that brings us directly to the February 2026 Nature study. Because that is exactly what they went looking for, right? The repair crew.
Exactly. The researchers from Northwestern UIC and the University of Washington decided that if the difference between a dementia patient and a SuperAger isn't the amount of damage, it must be the response to the damage. So they focused their attention on the hippocampus.
The memory center of the brain.
Right. And they asked a very controversial question. Are these old brains growing new neurons?
See, I thought that was settled science years ago. I remember learning in high school biology that you are born with all the neurons you will ever get. Once you hit adulthood, the factory closes down, and it's just a slow war of attrition until you die.
Oh, and millions of others were taught that. That was the absolute dogma for decades. It's a concept called adult neurogenesis, and it has been a vicious battleground in the neuroscience community. One camp said it absolutely stops after childhood. Another camp said no, it persists into adulthood.
Why was it such a debate? Couldn't they just look?
The problem was the methodology. Previous studies were really messy. They used chemical dyes or markers to try and identify new cells. But those markers could easily be misinterpreted. It was like trying to spot a specific person wearing a red hat in a very blurry satellite photo of a massive crowd. You could argue about what you were seeing all day.
So how did this new study finally clear up the picture?
They used a technology that simply wasn't viable or accurate enough 10 years ago. It's called single-nucleus RNA sequencing.
Okay, that sounds incredibly impressive, but break it down for us. Why single nucleus?
So brain tissue is incredibly dense. The cells are all tangled together with these long axons and dendrites. It's a dense jungle of wiring. If you try to separate whole, intact cells to study them, you often tear them apart, especially when you're working with aged, fragile tissue from an 80-year-old brain.
So you destroy the very thing you're trying to study.
Right. But the nucleus, the little command center inside the cell that contains the DNA, that structure is tough. It survives the extraction process. So instead of trying to untangle the whole jungle, they just extracted the individual nuclei.
Ah, clever. And the sequencing part?
They analyzed the RNA inside each individual nucleus. Now, DNA is your permanent genetic code, but RNA is the messenger. It tells you which genes are actively being used right now. It tells you what the cell is actually doing in real time.
I like to think of it like a library.
That's a great analogy.
Yeah.
Let's use that. If DNA is the library containing every possible book or instruction manual, RNA is the list of books that are currently checked out and being read.
So by looking at the RNA, they could look at a specific cell and say, you aren't just sitting there slowly dying. You are actively building parts to become a brand new neuron.
Precisely. They could see the exact genetic instructions being executed.
Yeah.
And they didn't just look at a few cells. They did this for 356,997 individual cells.
Over 350,000 individual cells. That is a staggering amount of data to process.
It is monumental.
Yeah.
And because of that scale and precision, the verdict is finally definitive. Adult hippocampal neurogenesis, the birth of new neurons in the adult memory center, is real. It persists well into old age. But here is the absolute headline of the paper. In SuperAgers, this process is on steroids.
Give me the comparison. What's the difference?
SuperAgers produce new neurons in the hippocampus at 2 times the rate of healthy, cognitively normal older adults.
Double the rate.
Double. It's a massive difference. And if you compare them to people with Alzheimer's dementia, the contrast is even starker. The patients with Alzheimer's showed negligible to almost zero new neuron production.
Okay, so let me make sure I'm synthesizing this right. In a typical Alzheimer's brain, the damage is piling up the trash in the streets, the tangled wires, and the factory that builds replacement parts is shut down.
Yes.
But in a SuperAger brain, even if that same exact damage is piling up, the factory is running a double shift to constantly replace the dying cells.
Exactly. It is a true resilience signature. The SuperAger brain is physically working harder to stay young. It is neuronally fertile.
I love that term, neuronally fertile. It paints such a vivid picture. Let's get into the actual mechanics of this factory, though, because the study used a really accessible analogy of the toddler and the teenager to describe these cells.
It's a very helpful way to visualize the lineage of a cell. In the subgranular zone of the hippocampus, which you can think of as the nursery of the brain, you start with neural stem cells. These are the babies.
And what defines a baby cell?
They are highly proliferative. They are dividing rapidly, and they have the potential to become different types of brain cells. They are pure potential.
Okay, so they are born in the nursery. Then what?
Then they differentiate into neuroblasts. These are the toddlers. They are mobile. They are physically migrating through the brain tissue, moving outward toward the areas where they are needed most.
And the teenagers?
Those are the immature neurons. At this stage, they have stopped dividing, and they are starting to extend their axons and dendrites. They are trying to make connections. They are a bit awkward. They aren't fully plugged into the neural grid yet, but they are maturing and trying to integrate into the network.
And the researchers found that the SuperAgers had more of all of these stages.
They had an incredibly robust pipeline. What they found in the Alzheimer's brains wasn't just a lack of the stem cells. It was a failure of maturation.
Meaning what exactly?
Meaning even if a few of those babies were born, they never survived long enough to become teenagers. They died off before they could plug into the grid. The environment was too toxic, or the internal instructions failed. But in SuperAgers, that entire pipeline from baby to teenager was wide open and flowing.
Okay, but this just pushes the mystery one level deeper for me. Why? Why is Gloria's brain at 84 running a factory shift that looks like a 25-year-old's while her neighbor's factory is boarded up and abandoned? Is it just that Gloria has better genes after all?
That is always the default assumption, right? It's in the genes. But this study suggests it is not the genes themselves. It's the access to the genes. And this brings us into the realm of epigenetics.
This is section 3 of our deep dive today, the librarian.
Right. So we talked about DNA as a massive library containing every instruction manual needed to build and repair a human body. Every single cell in your body has the exact same library. A skin cell has the instructions for making a neuron, and a neuron has the instructions for making skin.
But obviously a neuron needs different books than a skin cell.
Exactly. So who decides which books get read?
The librarian?
The librarian. That is the epigenetic machinery of the cell. In a physical library, books sit on shelves. In the nucleus of a cell, your DNA is wrapped around these spools called histones, and it's all packed into a dense structure called chromatin.
Chromatin.
Yes. Now, if the chromatin is tightly packed together, condensed, it is like the librarian has locked those specific books in a heavy steel vault. The instructions are there, but you physically cannot read them. If the chromatin is open and relaxed, we call that accessible. It's like the books are lying open on the table ready to be used.
So what did they find when they looked at the SuperAger libraries?
In the Alzheimer's brains, the librarian had essentially locked the doors to the neurogenesis section. The instructions for how to build a new neuron were still there in the DNA, but the chromatin was closed tight. In the SuperAgers, that chromatin remained wide open. It was what we call permissive.
So the fundamental difference isn't the genetic code itself, it's the lock on the code.
Exactly. And the researchers actually identified the specific keys to those locks. The study highlights specific transcription factors.
Transcription factors, let's define that for our listeners.
These are specialized proteins that bind to the DNA and basically tell it to start working. They are the switches. And they found uniquely high activity of factors like STAT3, STAT4, and STAT5 in the SuperAgers.
STAT3, it sounds very technical. What does STAT3 actually do in this context?
Think of STAT3 as the foreman on a construction site. Its primary job is to regulate cell survival and cell proliferation. If you are a baby neuron, a stem cell, you are very fragile. You need constant signals from your environment telling you don't die, keep dividing, keep growing.
And STAT3 provides that signal.
Yes, STAT3 is the foreman yelling, keep going.
In Alzheimer's brains.
The foreman is missing. The signal just isn't there.
Mm-hmm.
So the cells die before they can mature into toddlers or teenagers. In SuperAgers STAT3, along with other transcriptor factors like SOX6 and Neurod1, which help the baby cell figure out what specific type of neuron to become, are hyperactive. They are shouting instructions constantly.
This is where they talked about the irregulons, right?
Yes, irregulons are basically these gene regulatory networks. It's not just one switch, it's a whole master control board of switches that turn on together to drive a process. In Alzheimer's, the neurogenesis irregulons are powered down. In SuperAgers, the board is lit up like a Christmas tree.
Okay, so we have a factory that is open for business. We have a foreman who is awake and yelling instructions. But a factory needs a physical location. It needs an environment to operate in. You can't build delicate microchips in a toxic swamp.
Which brings us to the soil of the brain, the glial cells, and specifically the astrocytes.
Astrocytes. I feel like these guys get completely ignored in pop science. We always talk about neurons. Neurons
are definitely the rock stars. They get all the glory. But astrocytes are the roadies, the sound engineers, and the security detail. They actually outnumber neurons in the brain. And for a long time, scientists thought they were just structural glue. In fact, that is literally what glia means in Greek glue.
But they are doing much more than just holding things together.
Oh, absolutely. They are the gardeners of the brain.
The gardeners.
They actively maintain the microenvironment. They clean up cellular waste. They balance neurotransmitters. They provide nutrients directly to the neurons. Now, in typical aging, and especially in Alzheimer's disease, astrocytes undergo a massive change. They become reactive.
Reactive meaning they get inflamed.
Yes. They get irritated, they physically change their shape, they pull back their supportive branches, and they start pumping out inflammatory cytokines, which are basically toxic chemical signals.
So it is like a gardener getting angry and spraying toxic herbicide everywhere instead of watering the plants.
That is the perfect analogy. And so these new baby neurons are trying to grow in soil that has become actively toxic to them. It's no wonder they die.
But what about the SuperAgers? How do their gardeners behave?
In SuperAgers, the astrocytes don't get angry. They stay in a calm, homeostatic state. They maintain what the paper calls a pro-neurogenic profile. They continue to produce neurotrophic factors, brain fertilizer, that actively supports and nourishes the new neurons.
It is amazing how holistic this whole picture is. It isn't just one magic mechanism. It's the stem cells, the epigenetics, switches, the transcription factors, and the support cells all working in perfect concert to resist the decline.
It really is a systemic resilience. And it even extends to the wiring itself. The study specifically looked at the synapses, the actual physical connections between neurons in the CA1 region of the hippocampus.
And what happens there in a normal aging brain?
In Alzheimer's, those synaptic connections physically fray and snap. The communication lines go down. But in SuperAgers, the single nucleus sequencing showed that the genes responsible for building and maintaining those synapses are firing on all cylinders. The wiring remains thick and robust.
So that is the microscopic view. We have zoomed all the way in to the nucleus of a single cell. Now I want to zoom out for a minute. If I put a SuperAger in an MRI machine and look at the macrostructure, does their brain actually look different on the screen?
It absolutely does. And this is where we get into the structural hardware of the brain.
Usually the brain shrinks with age. Cortical thinning, that is just a sad fact of life, isn't it?
It is a standard feature of aging. Yes. By the time someone reaches 80, most people have lost significant volume in the cerebral cortex. That's the wrinkled outer layer where all our higher level thinking, processing, and memory happens. If you look at an MRI, it looks slightly shriveled with more cerebrospinal fluid filling the empty spaces where brain tissue used to be.
And the SuperAgers?
They show almost no thinning.
None.
Almost none. Their cortex looks remarkably plump. It looks like the cortex of a 50-year-old. But there is one specific area where they aren't just resisting shrinkage. They're actually superior to younger brains.
Wait, better than average, thicker than a younger person's.
Thicker than a 50-year-old's. It is a region called the anterior cingulate cortex, or the ACC.
Okay. Where exactly is the ACC and what does it do?
It sits deep inside the brain, just behind the frontal lobe. It sort of forms a collar around the corpus callosum, which is the bridge between the two hemispheres. The ACC is the heavy lifter of the brain's executive network. It is deeply involved in attention motivation, conflict resolution, error detection, and emotional regulation.
Error detection and motivation. That sounds a lot like tenacity or willpower.
Tenacity is a great word for it. It is the part of the brain that helps you push through when things are cognitively or physically hard. It is the willpower center. And in SuperAgers, this specific region is physically larger and more robust.
And earlier when we were prepping for this deep dive, you mentioned that this region is home to a very specific, very rare type of cell.
Yes, the von Economo neurons, also known as spindle neurons.
Why are they so special?
Well, first of all, they are physically huge compared to typical neurons. They're cigar-shaped and very long, and they are evolutionarily quite recent.
Meaning you don't find them in all animals.
Right. You don't find them in mice or rats. You only find them in humans. Great apes, elephants, whales, and dolphins.
The highly social animals.
Exactly. These neurons are thought to be the superhighways for rapid communication across vast areas of the large mammalian brain. They specifically link intuition, emotion, and social intelligence.
So SuperAgers have a higher density of these social neurons?
Significantly higher density. And this is the crucial bridge, isn't it? This connects the microscopic biology we have been discussing to actual human behavior.
Yeah.
Because if you have a massive, well-connected center for social intelligence and motivation that fundamentally drives how you interact with the world around you.
This is the perfect pivot point, because I know everyone listening right now is thinking, okay, I want the von Economo neurons, I want the open chromatin, I want the factory running double shifts. How do I get them? Do I just have to hope I have the right parents?
And here is the absolute best news of this entire deep dive. It is not just genetic luck.
Let's quantify that. How much of this is actually luck?
The broad consensus in the longevity and gerontology field right now is that your genetics account for maybe 20 to 25% of your lifespan and your cognitive health span.
Only 25%? That actually seems really low. I feel like people say, oh, my grandmother lived 95 with a sharp mind, so I just assumed I was set on autopilot.
Having good genes gives you a nice head start, sure. But 75% of how you age is driven by lifestyle, environment, and daily choices. The librarian, the epigenetic machinery that locks or unlocks the DNA, responds directly to what you do. You can force the librarian to keep those books open.
Okay, so let's talk about exactly how to do that. And I want to be really careful here because advice on brain health usually devolves into a listicle. Do Sudoku, eat blueberries, and take a walk. But the research we're looking at suggests that doing Sudoku might actually be a trap.
Sudoku is absolutely a trap if you are already good at it.
Explain that. Why is a brain puzzle a trap?
Because the brain is at its core an efficiency machine. Its primary biological goal is to conserve energy.
Yeah.
When you do something you have already mastered, like a crossword puzzle you have been doing every Sunday for 30 years, or playing a piece on the piano, by heart, your brain goes into autopilot.
It's just running a well-worn script?
Exactly. It is using highly established, thick neural pathways. It takes very little energy. And more importantly, it does not signal the brain that it needs to build new pathways. It does not trigger neurogenesis.
So comfortable is actually the enemy of brain health.
Comfortable is where your brain goes to sleep. To trigger neurogenesis, to wake up that STAT3 foreman and get the factory running, you need what researchers call an enriched environment. And the scientific literature triangulates 3 non-negotiable conditions for an environment to be truly enriched. It has to have novelty, complexity, and social connection.
Novel, complex, social.
You need all 3 interacting. If you sit alone in your room and learn a new language on an app on your phone, it is novel and it is complex, but it lacks the social pressure and engagement.
And if you just hang out with your friends at a cafe and gossip?
Then it is social, but it isn't complex or novel. It's just a routine.
So you need what you called earlier the vulnerable wobble.
The wobble. I love that term so much. It is describing a very specific biological state. Think about being on a bicycle for the first time. You are wobbly. You feel like you are going to fall. When you try to do something you are genuinely bad at, especially in front of other people, your brain goes into a state of high alert.
But cognitively, it feels terrible. It feels like embarrassment or frustration. Nobody likes feeling incompetent.
I know, but that terrible feeling is the exact biological signal you want. That frustration, that heat in your face, that is the brain releasing a flood of acetylcholine and norepinephrine. It is the chemical alarm bell that tells the brain we are failing at this task. We do not have the neural circuitry to do this. We need to adapt immediately, turn on the factory, build new neurons.
So if you aren't actively feeling a little stupid or out of your depth, you aren't growing.
Precisely. Let's look at the actual anecdotes from the people in this study because they illustrate this perfectly. There's a man named Raymond Watts. He is 81 years old, a retired postal worker.
Right? The K-pop fan?
Yes, it's such a great story. His teenage granddaughter is obsessed with K-pop music. So Raymond decided he wanted to learn to speak Korean so he could understand the lyrics with her. Now, Korean is linguistically an incredibly difficult language for a native English speaker to learn. The alphabet, the grammar, it's entirely different. And Raymond openly admits he has absolutely no aptitude for it. He says he is terrible.
But he doesn't just use an app, right? He goes to a class.
Right. He goes to a physical, in-person community college class. He sits next to a woman named Doris, who is 76. And they completely butcher the pronunciation together. They laugh about how bad they are. They apologize to the teacher.
That dynamic right there, the shared struggle, that is the key, isn't it?
It is vital. The social component amplifies the biological signal. Because he is interacting and feeling that slight social pressure, those huge von Economo neurons we talked about are lighting up. And that social challenge, combined with the complex cognitive challenge, causes BDNF, brain-derived neurotrophic factor, which is that fertilizer we mentioned, to just flood the hippocampus. If Raymond were just staring at a workbook alone at his kitchen table, the biological response would be a fraction of what it is in that classroom.
That makes so much sense. And then there's the other example from the paper, Gloria.
Gloria, yeah, she is 84. She decided to take up pottery.
Oh,
But she didn't just want to paint a pre-made mug. She joined a wheel-throwing class with people who are decades younger than her. She says she regularly makes creative disasters.
creative disasters. I love that framing.
It's perfect. She is dealing with a totally novel physical challenge. The wet clay, slipping the wheels, spinning, coordinating her hands in the pedal. And she is dealing with the social challenge of being the grandma in the room who can't even center the clay on the wheel. That is the ultimate sweet spot. She is living in the wobble.
It really effectively reframes how we think about aging and how we treat older adults. Because our instinct is usually to protect them from feeling incompetent. We see an older relative struggling with a new smartphone and we say, don't worry, give it to me, I'll just do it for you. Or we give them repetitive tasks they can easily succeed at to protect their pride.
And in doing so, we might literally be killing their brains with kindness. We are removing the very friction, the very stimulus that keeps the chromatin open. We need to completely normalize being a beginner at 80. We need to celebrate the struggle.
What about physical exercise? You said 75% is lifestyle. Does the physical body play a role in this factory? Do SuperAgers run marathons to keep the blood flowing?
Surprisingly, no. In fact, extreme high-intensity exercise in much older age can sometimes be counterproductive because it spikes cortisol and creates systemic inflammation. And remember, we want to avoid angry, inflamed astrocytes.
So what is the exercise prescription for a SuperAger brain?
It is all about consistency over intensity. The SuperAgers in the longitudinal study were rarely sedentary. They didn't sit in a recliner for 4 hours straight. They were gardeners, they were walkers, they were stair climbers.
Just constant low-level movement.
Exactly. It's about getting maybe 60 minutes of moderate physical engagement throughout the day. The goal is to keep the cardiovascular system efficiently delivering oxygen and nutrients to the factory without stressing the entire biological system into a state of inflammation.
Okay, so move constantly, but don't try to kill yourself at the gym.
Well,
And aggressively seek out social embarrassment and cognitive frustration.
that is a very unique way to put it, but yes, seek out socially supported challenge.
This research really completely changes how we have to look at the future of Alzheimer's treatment, doesn't it? This brings us to section 7 of our outline, which is reframing Alzheimer's.
It forces a massive paradigm shift.
Yeah.
Because for the last 30 years, the entire pharmaceutical industry has been obsessively focused on one thing, clearing the amyloid plaque. We actually have monoclonal antibody drugs now, like lecanemab, that are scientifically very good at binding to and removing amyloid from the brain.
But the real-world results of those drugs have been fairly mixed, right?
They are modest. Best case scenario, they slow the rate of cognitive decline by maybe 25 or 30%,
Yeah.
but they do not stop the disease, and they certainly do not reverse the memory loss.
And a SuperAger study explains why.
Exactly. It suggests we've been missing the entire other half of the biological equation.
Because if you clear the plaque but the factory is still closed down,
then you haven't fixed the problem. Going back to our analogy, you've successfully hired sweepers to clean up the trash in the streets, but the houses are still crumbling to the ground because there are no construction workers to rebuild them. We desperately need therapies that target the repair mechanism, not just the damage.
So pharmacologically, we're looking for drugs that mimic the actions of the librarian.
Yes, there is incredibly cutting-edge research happening right now where scientists are using brain organoids.
What is an organoid?
They're basically miniaturized, simplified brains grown in a petri dish. And they are growing these mini brains using stem cells derived from actual superagers.
That is wild. Growing a mini SuperAger brain in a lab.
It is the absolute frontier of neuroscience. They use these organoids to test hundreds of thousands of drug compounds. They are looking for molecules that target the STAT pathways we talked about. Can we find a chemical that forces the locked chromatin to open back up? Can we invent a pill that wakes up the STAT3 foreman in an 80-year-old with early-stage Alzheimer's?
That would be the holy grail.
It would change the world. But until we have that pharmaceutical pill, the societal pill is the only one we have. And honestly, this research is a scathing critique of how we currently handle aging as a modern society.
You mean warehousing the elderly?
Right. We tend to segregate older adults into these specialized care facilities where absolutely everything is done for them. Meals are prepared on a schedule. Their day is perfectly structured. Risks are entirely minimized. It is essentially a zero-wobble environment.
We are designing environments that are perfectly physically safe, but biologically completely stagnant.
We are effectively boarding up the windows of their neurogenesis factory for them. If we want to implement the lessons of the SuperAgers, we need radically different social structures. We need intergenerational spaces. We need environments where an 80-year-old is challenged to learn a new technology alongside a 20-year-old, not just have it explained to them like a child. We need spaces where older adults are genuinely needed to solve complex problems.
Giving them purpose and real friction.
It sounds like soft psychological advice. But as we've seen today, the single-nucleus RNA sequencing proves that this soft advice is driven by very hard biological hardware.
Let's bring this all together. We have covered a massive amount of ground today. We started with the central paradox, the dirty brain that works perfectly.
Right, and we found that the solution to that mystery wasn't resistance. It wasn't about possessing a magical Teflon brain that repelled aging. It was resilience. It was about having a brain that could take a profound biological punch and just keep growing anyway.
We zoomed all the way into the cellular factory and saw the neurogenesis. We saw the baby, the toddler, and the teenager neurons being pumped out at double the normal rate.
We met the librarian, the epigenetic machinery that is keeping the instruction manuals open, and we saw the gardeners, the astrocytes, keeping the cellular soil calm and fertile instead of toxic and inflamed.
And most importantly, we learned that the key to keeping that entire complex system running isn't just winning the genetic lottery. It is the active daily willingness to embrace the wobble.
The profound willingness to be a beginner again and again.
So we always like to leave our listeners with a challenge or a provocation at the end of a deep dive. And based on the science today, I think the challenge is pretty clear.
I agree. It writes itself.
We so often think of aging as this defensive process of holding on. We spend our later years holding on to our past memories, holding on to our career status, holding on to our image of competence. We are terrified of losing our grip on who we were.
But the science, the actual molecular biology of the brain, suggests that the only true way to hold on is actually to let go.
Let go of the need to be the expert in the room. Let go of the warm comfort of your Sunday routine.
Embrace the awkwardness and the confusion, because that confusion is literally the feeling of your brain growing.
So here is your specific mission for the week. I do not care how old you are listening to this. You could be 25, 50, or 85. I want you to find one thing that you are absolutely undeniably terrible at.
And find a group of people to go be terrible with.
Sign up for the weird class. Join the club where you don't know the rules. Go find your wobble and keep that factory running. Thanks for diving deep with us today.